Yes. Yes, I just started the recording. Yeah, so to bring all the audience to all everyone to at the same stage so that everyone gets an idea about the basics of bioinformatics, and then we will look at for the varied applications. So then we will look at the some of the alignment sequence alignment algorithms like Middleman-Jones algorithm or Smith-Waterman algorithm, the BLAST, which is extensively used worldwide for various bioinfo applications in molecular biology, and then we'll just get upon with the if our time permits to just look at the PDB, the protein visualization, just to have a demonstration kind of thing. We will have a detailed session on this protein visualization and the visualization tools like Chimera in the later in the coming weeks. And so, so before that, I would like to ask you all. So, how many of you know this lady, or how many of you seen her picture or know about anything? So those who know, you can chat, type in the chat box. Let us actually, I want to make this session interactive so that we can learn from both ways, as well as not a kind of lecture thing, but a informative session. So how many of you know this one? So you can unmute yourself and tell, or otherwise don't start speaking all at once, or can raise hands, or you can write in the chat box also. So how many of you know without obviously referring to any Google sources? Is someone telling anything?
Margaret Day.
Sorry, who is this? Kindly tell your name and yeah. I can only hear the background, so you can mute and tell it clearly.
This is Srinity.
Okay. Yeah. So what was your answer?
Margaret Day.
Okay, fine. So good. So you know. So yes, correctly. This is Margaret Deva. So this can be said that she is one of the ones who started or inspired or began the field of bioinformatics, with all the sequences getting generated each day, with all the technologies coming up each day. We all know that how much it is required. So we can just think of or appreciate her vision or her thinking at that age, at that time, and that too was 1966. So this is was the first kind of sequence database that we come to know about. So that is the Atlas of Protein Sequence and Structure at 1966. So here, if you go and search and read this paper, so you can see that what was there were only around seven or some sequences, those known protein sequences, and their sequences of tRNAs and all were kept, but just please be muted. Just ask Gail when I speak, otherwise it will be disturbing. Yeah, so yeah, so for them, so from this on, the thing just I want to give you a glimpse of this history, because that you will feel or you can appreciate that how it inspired the whole field, and how it is getting utilized every day, and now how important has become the field of bioinformatics and computational biology is it. The thing is that the history always inspires the thing and helps the people to think ahead, much ahead. So see, so if we can get a timeline of the field of bioinformatics, how it has started. So after 1966, Atlas of Protein Structure and Sequence by Margaret Daya, so we get the from Brookhaven National Laboratory, the PDB, Protein Database Structure, and it was established in 1971. So it only inspired that we should have come with someone which contains the protein structure as well. And it originally contained only seven structures, and you can just think of that today it contains more than 30,000 structures. PDB, and it's being widely used, deployed for your drug repurposing based approaches, for your identifying targets, would know the protein structures, and in every field. So this was from 1970s we get. In 1971 came the first sequence alignment algorithm. So you can just think of that the DNA sequences, the protein sequences are all, we cannot interpret anything with a single sequence. But if we can align them, then we can identify the similarities, the dissimilarities, the evolutionary trees, the conservation of sequences, domains, motifs, everything we can understand. We can understand why, from starting from the evolutionary origin, you can just understand that now the variants of the COVID-19 also is being utilized by this alignment algorithm to identify the genes that part of the nucleotide sequences responsible in cancer. So this how it came up. So this is the Gittelman Rules paper, which told that the structure similarities in the amino acid sequence of two proteins. Next came up the first. Then after that, now the alignment was done. Now that there was the need to for the identifying the protein structure prediction algorithm, which is quite much needed now also, because we don't have the structure of all the proteins, but it becomes very much important because without proper structure, a protein cannot function. So I am also just trying to give a basic things about, because there is some engineering background students also. So protein can only function well if the structure is known. So for that, protein structure prediction algorithm was done by Chow and Fasman. Then came the gene bank, which contains all the comprehensive nucleic acid sequences from all over the world. And then came the blast, basic local alignment search tool by Stephen Alstrom and FASTA alignment tool. These all became necessary because if the two known protein sequences are there, then Needleman knows or Smith Waterman, we will come to it in the later stages, can be useful. But if we want to perform an unknown DNA sequence matched with all the sequences present in a database, then came all the inventions. Now, biological databases are knowledge discovery. So, for instance, all other databases, if something you put up, that the information is okay. But in a biological database, if an unknown DNA is sequenced, then we need to find out its evolutionary origin. We found out its conserved region, conserved sequences. Is there any mutations are present or not? So biological databases needs to be a knowledge discovery. You all might know that biological databases are generally only of it's only normal databases are like relational databases are there, object oriented databases are there, but biological databases needs to be much more powerful and interlinked type kind of databases where you have to interpret things also, not only the information, but you have to interpret it value also. So there are three types of databases in biological databases generally. There is primary database, secondary database, and specialized database. So what these three things mean? So now after this thing, if you understand this term, then you will try to identify whenever you go to some database to search for your sequence or something, then you will be able to understand that what kind of databases are there. So primary databases are basically those databases that contains only the original information. They mainly provide us the original information. Secondary databases are those that curates the data of the primary databases and gives the provides the storehouse of those databases. Like you can say, like Swiss prod is a kind of a secondary database. And specialized databases are those that are specialized for a particular organism or a particular region of DNA. So, like fly-based, it contains of all the interest of the fly and not other. So these are a kind of specialized databases. So nucleotide sequences like GenBank, EMBL databases, and DTBJ, DNA database of Japan. These are mostly primarily a kind of primary database where sequences are there. And the thing is that a correlation, there's extensive correlation exists among all those three because the data that is stored in the gene bank and the EMBL database or DTBJ will go and get circulated, get exchanged within 24 hours in all the three servers and it will get updated. So it's a it contains all. So anywhere we go, means anywhere you submit, this is required for the research purpose and all publicly available repositories. So anywhere you go, anywhere you submit your data, which you, for example, if you sequence an unknown organism or find an unknown organism, you will find it all those three. So there's exists a very good collaboration. Then the protein structure sequence is there, where you can find the structure is the PDB. So now some major biological databases that are being used very commonly is that DDBJ, MBL, Untrace, Expasy. So we will try to go visit this in different days and different contexts. We will like to see these databases, how they function, how they can help us. But these are mainly the used databases like Flybase, these HIV databases. Now you can understand because specialized databases only those who are interested for this kind of research and won't go in other like PubMed. So PubMed is also for the literature based it's a database where you can curate all the biomedical literature that is present. So these are the some major biological databases. So now another thing is that the biological databases needs a very good robust annotation information. Otherwise, otherwise there are sequences being deposited every day for the same genes. There are different kinds of sequences coming up for different kinds of sequences. So it all creates a very much redundancy in these databases. So it will be become very it becomes very difficult to come up. So you cannot identify. You become confused that which one to take, which one not to take. So the biological databases need something identifiers or uniqueness to identify that it is a particular gene or particular way you are going. So I will look at it how it is important to identify some identifiers so that we get the exact one we are treating. When biological databases suffer from redundancy, they expect from NCBI to try to help it out. Another thing is that if the annotations are not correct, then for the same gene there will be different sequences and all types of uh, complexities will arise. So now another thing is that in the biological database it is required is a sequence to be retrieved. So you need to retrieve the sequences, you need to retrieve all those. So NCBI uses Anthrace and European Bioinformatics Institute to sequence SRS program to for performing the sequence retrieval. Now, so everyone, if you can go and in the gene bank. So let us go at the gene bank and sequence. So we know that I told just now that it is very much important for an identifier. If I don't have this identifier, and if I say, let us look at first what is this identifier. So if you all can also do on side by side. So I'm also pasting it in the chat box, the link. If we go to a nucleotide and paste the accession sequence, yeah. So have you got this page, everyone?
Sir, I
Just take.
cannot use chat box, sir, because it is showing that it is available for only team members, sir.
Okay. So no one is able to access.
Yes, sir.
So people who have joined as a guest can't access the chat box.
Okay, so just type it from the screen. I'll check it today that what settings something can be done. Okay, so age I'm repeating also A015990.1. Yeah, so if you everyone is on the same page, then let us look at the format. The file formats are very much important in the its databases to have a uniform identifier and uniform identification of all this. So let us just look at that. What does it present and what are those? What does it mean? Okay, so this is the if you can see in the page. So this is the number accession number, that is A01590. Okay, so if 423 base pairs means it is the length of the DNA sequence, and the DNA is linear, and it has been given that 3rd July 2002. So it is an human hemoglobin A alpha chain, and its version is 1.1. Point 1 means if there is some changes made to it, so accession remains same for a particular gene, but with the GI number and with its version, it gets updated if any update comes. So source is Homo sapiens human. So organism is Homo sapiens, from where it is the source. So bases 1 to 423, it is a 423 base pair long DNA sequence. So this is a journal, this is where it has been published. Now it is CTS. So CTS means the region of those particular DNA sequence which codes for the protein. So this is the region only that it is the protein that gets coded from this from the genomic DNA. So this is the translation. After translation, it given the protein. So codon starts from 1 and ends at 423. Means all the 423 sequences present here codes for the protein. And this is after the translation, after the three translation. So this is the and this is the origin. And always remember in that NCBI in the gene bank reference format, it ends with double slash. Okay, so this is the total to understand what is present in this file format of the gene bank. So in this way, it describes about the human, it contains the sequence as well as describes the human hemoglobin A alpha chain. So everyone is has understood, right? Hopefully you are doing also. Okay, so now another file format is the FASTA format. So if you go and look here, so there will be a written as a FASTA. So if we click that, so we will get a FASTA format. So this format has been used very much for various sequence alignment and various tools. So for FASTA format, you remember that it always starts with a greater than sign. It will start with a greater than sign, have the accession number and a short description. So if you are want to construct anytime your own FASTA database for your own customized work, so always remember to that FASTA format will start from the greater than sign and it will contain the accession number and it will have the short description followed by the sequences. So this is the FASTA format. And if we go to this, it is the GenBank format. Fine. So we can retrieve this FASTA format. We can just save it as a in our text as a dot FASTA, or we can download it. Now, so now another thing is that if you want to change your file format, if you have a FASTA format and you want to check for the GenBank format or anything else, so there is a tool from Emboss Secret. So let us go there. Just type Emboss Secret. I could have not pasting the in the chat link. So everyone has gone to this.
Yes, sir.
Just yeah. So just raise your hand just to show that you see all of with me now. Yeah. Okay. Okay. Fine. Okay. So you can lower your hand. So let us look at it. So here is that we need to change our from file format from one to one. So here we will set a interface of protein sequences in any supported format. So what are the supported formats? Right. So supported formats, the FASTA format is mostly supported in every tool. Okay. So what are those? Here you can, if you go there, you can see a lot of formats, everything, protein, DNA, RNA, everything. So, so for us, so we will take this FASTA format. We will just copy it. So just remember that the FASTA format contains with the that the FASTA format contains with the greater than sign, short description, accession number, and the sequence. Now we want to convert it to if the input format is not unknown format, so you can see that this many formats are you can give, or this many formats exist in total in bioinformatics. So we will choose here FASTA format including NCBI style ID. Output format, what kind of format we want. So we can give it like if you want to see GenBank entry format, which format we just show that it contains a local definition and all. So we want from FASTA to change into that. So what we will do, we will just now submit. So it will go to the web service, it will okay. So we have got right the same locus, the definition and the origin. It will not give the CDS and all, but we can if some tool wants, if you have a FASTA format and some tool wants you to give the input file in a GenBank format, then this will work. The main thing is that to keep in mind that the GenBank format always ends with double backslashes, whereas the FASTA format always starts with a greater than sign. So everyone has got this result. So you all have raised hand that you have got the result. Okay, no question, right? If you have any question, you can unmute. Anyone can ask. Till now, no difficulties, right? Okay, so good. So no one is facing any difficulty, right? So you can go ahead. So, so just you can appreciate the fact that there are so many databases, like see, so many file formats are there, so many sequences are there coming up, so many organisms are there, so many groups are working, so many things are coming up daily at every time, every moment, every second. So the databases need to be a very much have a very interconnected database and the network should be very strong to for one file format for one to understand and for one to analyze and interpret. For example, if a nucleotide sequence is submitted in a gene bank, to it should be understood by the software that translates it to protein sequences to predict the protein coding genes. So everything. So the data biological databases are very much interconnected, and it is a combination of all relational, object oriented, of all kinds of databases. It's a combination of this. So it helps in storing the information as well as in the collecting information as well as in the knowledge to interpret the result. So now let us go to the sequence alignment. Sequence alignment, you all know that this DNA to sequence it or the sequencing technology started from standard sequencing to the today's next generation sequencing or the nanopore sequencing. So these are just stretches of ATGC. So these are nothing. So if we have only one sequence contents of any length of the sequence, for instance, if we sequence any human being having billions of base pairs, so we cannot interpret anything. We don't know that it is correct, it is wrong, how what is that, what to interpret, how to interpret. But it should be interpreted with something else. It should be compared with something else. That yes, it is. Is it the same? How much is the same? How much it is different? So, for instance, if we compare humans and chimpanzees DNA sequence, so we can see that we both share around 99%, 98.8% of our DNA sequences are same. But just think that how chimpanzees are different and how we are different. So in this case, it cannot be possible to know or to understand without having comparing the sequences. And just think that if we are given 5 or 10 sequences long, the time you will take to do it manually. If for a human being, if it is a billion sequences, then the computational power is required, how much the computational power is required. So here comes the bioinformatics or computational tools that plays a major role in analyzing and interpreting this huge amount of data or the data set, without which the sequencing or the data generation is important, but interpretation becomes much more time consuming, cumbersome if their computational tools and the alignments doesn't come. So this sequence alignment algorithms has also a history where different kinds of algorithms have been developed by and deployed to identify. But the thing is that the two things need to be kept in mind while designing an alignment. And I think that this alignment algorithm can still be further improvised and improved because it the data is getting generated in a much rapid amount now, and we still need better versions of alignment. So that first alignment uh, so before coming that see before alignment was developed as a to perform a global alignment. The global alignment is to look at the entire region, the sequence one and sequence, for instance here. So it will the algorithm will run to identify that the sequence is it is matching or not, it is matching or not, it is matching or not over the entire region. So this is a kind of global sequence alignment. And the local sequence alignment is that it will perform only in the small region where the maximum matching is there, and it will not go for the entire region. So anyone has a question? Shinjana here, Vishani, you all have raised your hand.
No, sorry, sir. That was a mistake. I did not understand. Sorry.
Okay. And Vishani?
So
Okay.
it was a mistake.
Okay. Fine. So this is now this sequence with the sequence alignment, we can note about some terms like sequence homology. That these two sequences are homologous. Homologous means that these sequences have been derived, have some evolutionary origin. That these two sequences come from the same origin. This is the sequence homology. And the sequence similarity, how much the sequences are similar. Sequences identity. So now there are so just think so about the sequence similarity only we will we conclude about the homology of the sequence. But the sequence two sequences can be similar but are not belong to the same origin. So these are not homologous sequence. Another is that if the in the protein sequences, for instance, if you compare and find out that in the DNA sequences, though there were differences, but in when it was translated, it contains the same amino acid. So that means the sequence are identical. So here it is the few terms which you need to keep in mind. So now going on, so sequence alignment first was came was the dot matrix approach. What is the dot matrix approach? In this dot matrix approach, if you can see, so first kind of sequence alignment which has been developed was the dot. Here you will write the sequence. Here you will write the sequence, and you will if G to G matches, you will put a dot. If G to it doesn't match, you will not put a dot. If T to T matches, you will put a dot. If T to T we have matches, you will put a dot. And in this way, you will find out where you can go diagonally, you can cut. And in this way, you will find out the matching. So here, let us go and check it. So it is, you can understand that it is very difficult for a large number of sequences. So that's why it has been improved and further new algorithms have been developed. So today also, if any algorithm is still not being fulfilling our needs and requirements, it will definitely there is a chance of improvement. So let us go to this emboss tool dot matcher where we can check out that how it is working. Type emboss dot match. Okay, so yeah, Cyan.
So actually, I just want to request you that can we get the slides after the class?
So I'll talk with this Sarah, this other TS about it. Okay.
Okay, sir. Thank you.
So everyone has reached dot matcher. The others are facing any difficulty? Okay, fine. Then we will try to perform this. So we will take this sequence only. Enter the data manually, type here, so it just copy pasted the sequence. Now we have to check with another sequence, right? So I'll just make it. So just made it a beta chain. So you can look at it that how we can search it by name also, and how fast it is pretty fast with all this so many sequences. So just checking the beta chain. So just remember that we are taking it in a faster format. So I think you all can now realize that which format it is. The alpha chain, beta chain. The window side. So we are just not doing it anything, just taking it by default what is there. So you all can play with it and can give different threshold and can look out that how it's working. So we'll run the dot method to see. So if we give this title and all, so this is the sequence, this is the sequence, and where it has been merged, we can get this by diagonally. But here it is only of 400. How many sequences are there? Only 400 something sequences are there. This happens, and if we know both the sequences, then we are performing. So you all can understand. So if we give a human sequence, it won't be possible. The server won't allow you to do. So this sequence, this thing is not possible. But just I want to just to give a idea how it evolved, so that you can understand how the necessity came and how the algorithms got evolved and developed, and where we stand now and how improvement is required. Correct. So this is the embossed secret. So people can use this and can identify using various sequences and all. So this is the dot method. So everyone has done it, right? Or anyone has any stuff that somewhere?
So can I ask you something?
Yeah, yeah.
So what happens if we increase or decrease the threshold? I just did not get it. During the dot matching, when we after we put the faster sequences, what if
Ah,
we increase or decrease the threshold? What is going to happen?
one second. It will show you what happens. See here, many things. The window size threshold matrix. I have not come here yet. Till the matrix, I will tell what is this matrix here. The threshold is the. Let me just make it 50. Then submit it. See, so this is the score. So now you understand, right? The density it has become. Correct. So with the scores, with only two sequences, if it is matching, it is
Yes,
making
sir.
the cross. It is making the cross. Correct. So if once you increase the threshold, it is the more stringent. So once you increase the threshold, the number of sequences when it the diagonal gets, then only it will form the cross. Correct. Clear, you're right. So that's why I told you if
Sir,
you just yeah,
sir, your voice is breaking, sir. Is that is only for me?
that can say is my voice breaking or I'm audible, right, to everyone? Just confirm it, anyone else.
Yes, sir.
Yes, you are perfectly audible.
Okay, so I think you can check your connection once. Yeah, so this
Okay,
is
sir.
the thing. So threshold, if you are looking for a very divergent kind of sequence, that you know that these sequences are different, then you will go for a performing lowering the threshold. But if it is a very closely related sequence, you will increase the threshold. So, any with your question, you will do that accordingly. Okay, so now, okay, so you will ask questions you can, okay, if you have any doubt, you can ask in between. So, after the dot matrix approach came the dynamic programming alignment, Middleman Wunsch algorithm. So middleman rules is so see here two things are important. One is the dynamic programming and this one is the global alignment. So middleman rules is a global alignment means it takes the entire sequence of the data. And what is the meaning of the dynamic programming? So dynamic programming means if there are two sequences are there and if it in a row, so it will go row by row. Means every sequence it will try to match with every other sequence. Every sequence it will be matching with every other sequence. So if there are 10 sequences and 10 sequences, so one sequence it will try to match with 10 sequences. Again, another sequence will try to match with the 10 sequences. In this way, this will go. So just think that how much exhaustiveness or how much power is required. So if you want to do for a thousand sequence or for a hundred sequence is a thousand sequence, and then for a million and then for a billion sequences, how much it will go. So it is also not feasible to do. But still, middleman was the first time try to perform some kind of alignment strategy. So let us look at it how dynamically how we can we can look at it, and also it introduces the penalties and all. So I don't know how much if you all have here in your courses this the middleman owns in bioinformatics if you all are having in your regular courses. So but here you can find it interactively how it is going without pen and paper. You can if you can just include this score and all, we can find out it. So if visible, my screen means everyone has reached here, right? Okay. So see, here it is written is that sequence one. So now if I increase the sequence length, see just see that how score is moving. Fine. So see that how it moves, it will match with the G with the Z. So score from diagonal cell is 0 plus 1. It will be 0 plus 1 due to a match. If it is not, then score from upper cell minus 2 plus minus 2 gap score. For gap it is minus 2 and for mismatch it is minus 1. The next cell what will be? The next cell will be minus 2 due to a mismatch between T and G. It is a mismatch, so it will be minus 2 plus minus 1 due to a mismatch. Fine. So it will be score is minus 3. Score from a diagonal cell will be -2 plus -1, and score from the upper cell will be -2 plus -2. So now in this way it will go on to calculate the score for each cell. So just think that if you are given such a this 1, 2, 3, 4, 14 length sequences, so how much you have to calculate for each score. So if it is 100 sequences, then the program needs to run for each score and to calculate the score and finally trace it back diagonally. Okay, so this is the Middleman-Wunsch algorithm where you can also change the match score and the mismatch score and the gap score accordingly, and it will accordingly give the uh, Middleman means the alignment. So this was the Middleman alignment with the dynamic programming for the global region. It will take the global region. Okay, so another dynamic programming is there. So just keep into things that the dynamic programming was used by this for Needleman wounds and Smith Waterman. Needleman wounds perform the global alignment and Smith Waterman perform the local alignment. So now going back, yeah, stay clear, right, everyone?
Yes, sir.
So here the dynamic programming alignment for local alignment is the Smith Waterman alignment. So here we will follow these two things. Let us look at Smith Waterman itself. Okay, so here is this algorithm, and just look at it. So Smith Waterman here, what major difference you can find out that it will not trace from the diagonally from the last step. It will do it from where it is the maximum score it will get and maximum where it can go. Okay, so 2, 1, 0. Correct. So the maximum score it can go. So from here it can go maximum here. So it took this. So Smith, so it is performed. It will check for any local that which from where is the highest score and from where selection goes. So this is the Smith Waterman local alignment. Now let us do one on this server. It's the local alignment web server. So Smith Waterman is using this. There has been a web server developed. So let us look at this. Okay. So again we will paste the sequence. So everyone please go to this server. Okay. So it is, I will wait after pasting the sequence. I just ask you.
Sir,
Yeah.
sir, in needle man, which we have calculated score, no, sir.
Ah. Yes.
How we calculate that score, sir? What's the logic behind it, sir?
The logic is that see, match score is +4, mismatch score is -1, and gap score is 2. So if you go to this website and see that here it will show that why it has given +4 because it is a match between G2 3 and here nothing is there. So 4 plus 0. Okay, so it is 4. Correct.
Yes, sir.
So now here if you go, why it has been given as 2, it will show the score from the side well 4, okay, plus -2. The gap score, it will give the 2. So winning, it will calculate from See, the thing is that if you not read about it, so this is the cell it is taking, so it will calculate from upper cell, this cell as well as this cell. Okay, so here is the three scores are given. Okay, just to for clarification. So those who don't know about the determinant algorithm, so we have to score calculate from all the three cells: diagonal cell, upper cell, lower cell, and the winning score means the highest score will be put at its score. Okay, so score from the diagonal cell is 4 plus minus 1, 3 due to a mismatch between T and A. Score from side wall is 2 plus minus 2, the gap score is 0. And the score from the upper cell is also 0. So maximum score is 3. So for that thing, the cell is the score of that particular cell will be 3. So just think. So firstly you have to calculate for each cell you have to calculate three values and then have to select the maximum score. Fine. So this is the development algorithm. So if you go here, every cell you will find out that how to do. So you all can do one thing. You all can take a sequence, practice it in hand, and then can verify it here that it is coming correct or not. And those who will appear in exams and all those are regular courses, so they can do this because these things comes in exams also. So this is how the scores are calculated. Correct. So if you change here, it will change here. The mismatch score and the gap score. Clear, right?
Yes, sir. Clear, sir. Thank you, sir.
So we'll take this. So I'm not going to do change anything. Okay. So gap opening means that when the gap opens in a sequence, for instance, a sequence is coming, but if a gap opens, then the penalty will be given as minus 12. For each extension, it will be minus 2 gap if for each where there is a gap and it extends, so it will score will start giving minus 2. So minus if there are two gaps, so for opening it will be minus 12, and then for extension minus 2 it will be minus 14. So this is there. So I'm not changing anything, just to check out how the result comes. So you all can change all this and can look at it. Okay, so here we will find the alignment file. Okay, so this is a local alignment it has performed, and see it has given those places where the alignment has come. So have you all got this result? Yes, so just raise your hand if you all have got. Yeah, that's facing any problem. Okay, so this is how it performs and give us. So there are various scores they have given, like what a man got and all. It gets the score. So accordingly, you can find out for 39 points for various thresholds and all. So you can find out. So the C also in the human hemoglobin C just the thing that in the both having from the same protein, it has a different sequences. So how diverse the things are and how important these things are to know about it. Okay, fine. So now, if there is no question, then going ahead. Okay, so there you all find out that there are very matrices source are there, like CAM, Blossom. So what are these? So CAM was given by Margaret Dayhoff. She developed it. So it is that a CAM is called as point accepted mutations. So what is a PAM unit? So if you can see, a PAM unit is that if it is one, PAM number is one. So the observed mutation rate will be one, and the sequence will be 99% identical. PAM 0 means 100% identical, and PAM 250, which is mainly used for to identify the diverse sequences. So mutation rate will be 80% and sequence identity will be 20%. So now accordingly, if you are trying to align a closely related sequence or a very diverse related sequences, then you can accordingly set up your scoring metric, PAM 250 or PAM 30 or PAM 80 or PAM 100. So PAM means this point accepted. So the rate, the mutation rate that you are allowing. Okay, so now another thing is the blossom. If you have heard doing blast, you will find out blossom 62, blossom, various things are there. So blossom is that is a blocks of the content which is remains conserved. So those the sequences identity that are made, the sequences the sequences identity from which that it is made up of the alignment that consists of the blossom 50 or blossom 62. So here it is the opposite. If it is lower, blossom number is lower, then means the sequences are diverse that it comes from a very different related species. And if PAM number is greater, then it comes from a different species or different highly. But if the closely related sequences, then the PAM number should be less and blossom number should be high. So keep these things in mind while giving the parameters for the matrix. Yeah. So database searching here is that heuristic database searching is that kind of searching. Means for instance, you all saw that we are giving two sequences and then it is matching in the both in the middleman nodes as well as the mid waterman or the aligned web server that is kind of that uses the local alignment feature. We need to provide both the sequences. But if we have one unknown DNA sequence, if you have cultivated some culture or have you identified some organism and sequenced those, and you want, and you all know that the NCBI contains such a huge amount of database, amount of sequences. If you want to identify that which sequence is there, or which it is matching, or what is the evolutionary tree, what we'll do, we will just paste the sequence there and perform the databases to identify. So BLAST is that type of tool which performs this, and BLAST is having uses k couple method where k is you can say k means the number of words. So for BLAST generally it uses three. It searches three key keywords in that sequence and identifies. So and also BLAST is different type of BLAST. So blast 10, if you have a perform want to perform both the nucleotide searches, then it is blast 10. Blast X is for the if you want to translate the nucleotide sequences and search it against a protein sequence, then blast X. Blast P is there for two comparing two protein sequences. There is a T blast chain. Blast chain where you give the protein sequences and then you translate the nucleotide sequences and search it in against the nucleotide database. Similarly, T-BLAST6 for the protein sequence searching against the nucleotide sequences. So this is how, for instance, for T-BLAST10, if you take a protein sequences for nucleotide and you want to identify some protein coding genes, that which genes are responsible, you will perform T-BLAST10. So another database algorithm is that FASTA is fast solve. So let us go first. We'll perform blast, and they will understand. Another thing is that in the blast to know about is the p value and e value. E value are the probability it says that the sequence that has been matched by chance. Okay, so the e value corresponds to it. So the lower the e value, the we are more confident of the alignment, and it is not by chance. So lower, so we have to if the last value comes to be remember the e value to be less. So let us perform just the last to see. Okay, go to Instagram blast. So you can see that we can paste their accession number, gene ID, or the FASTA sequences, and here it contains all the what kind of we want to do. So let us just put this FASTA sequence there. Okay, so it is the nucleotide. The algorithm parameters. If you can see here, we can change the match mismatch score, gap cost. Okay, so you can optimize for highly similar sequences or what. You can give the or exclude some organisms if you want that you know that this organism is not there. So roll results in a new window and perform the blast. The wallet performing, right? Yeah. So how many of you got this result page? Just be little bit quick here. Yeah, okay, fine. So let us look at the values we have got. So we can see that the maximum score, the total score. Okay, so Homo sapiens sapiens is the mRNA hemoglobin alpha chain, it has got matched with it, and the e value is 0, percentage identity 100%. Means it has matched perfectly with that. So here we don't need to worry about it because we have given that sequence only that will be getting matched. So here percentage identity and e value is 0. So it's for ideal where we get the 100%, uh, we get a 100% matching, but it doesn't happen every time. So then we have to look at for the e values to choose upon which has occurred. It is not random and it is highly conserved and highly that it is very much what you say, it is very much it is not random. It has been very much confident of the score. Okay, so similarly we can have the pasta. Pasta is that it uses the hashing algorithm. Yeah. Okay. So here also you can using the program faster you can perform the learning.
Sir,
Yeah.
sir, could you please explain blast algorithm parameters, sir?
Okay. So I think one thing we can do is that you can just look at this faster also alignment also. Since we are running, we have run out of time. So what we can do is that we can start from here only from last and first algorithm parameters and then go to ubiquitous PDP and then go to that. So I will start a time previous next week onwards, and we will then discuss it. Is it fine?
Yes, sir.
Okay, everyone. So, does anyone have any doubt for today's regarding today's all the things that we covered? So, if it is done, then we can conclude the meeting today, and we can start from here. Okay. Okay. Fine. Bye. Thank you all.
Thank you, sir.
Thank you, sir.
Thank you.
Thank
Thank
you,
you,
sir.
sir.
Thank you, sir.